Allison Justice. Dr. Allison Justice, it's so nice to meet you and to have you here on MyFlora Fridays. We were just talking about how we've never actually physically crossed paths, although I think we've been running in the same circle for 3 or 4 years now.
For
So
sure.
obviously, I don't know that much about you. I know a few things, but you know who you
Sure.
are, and
Yeah,
better than me. So can you introduce yourself? Tell us who you are, what you do, and what brought you to the cannabis space?
absolutely. Well, that is a very intensive question. I'm Dr. Allison Justice. I'm very excited to be here to speak with you today. Like you said, we've been running in the same circles for a while, and I'm excited to sit down and talk to you and share it with everybody what we discuss. My background is truly in greenhouse production ornamentals. That's what all my grad work was in, and that's what a couple of years after grad school, down the road it took me is working with big box stores. So poinsettias and petunias and mums, so flowering crops, which coincidentally works out pretty well. They've got a lot of the same thing going on from photo period to massive amounts of vegetative cuttings for reproduction, so that set me up pretty well as a background prior to getting into cannabis. Being from South Carolina and graduating quite a few years back prior to even hemp being federally legal, I never necessarily thought cannabis was in my future. I appreciated it as a product, and used it myself. I just didn't didn't think it could be a career choice. Um, but the way I actually got into it was after graduation. As I said, I was doing some consulting for greenhouses, mainly IPM, and started getting some calls from cannabis companies for IPM, a lot of the same problems. And at first, I didn't necessarily know what to think, because any of those opportunities would, literally be on the other side of the US. But I was young and excited about everything in life. So I said, well, let's try it. Let's see what happens. And, took a few jobs and absolutely fell in love with the plant. Fell in love as a researcher with the opportunity of so much needing to be dug into, and still will be needed to be dug into well past my lifetime. So being a part of that was super exciting. So I took a job in San Diego, California, and I stayed out there as VP of cultivation. We had indoor, outdoor, greenhouse production, and that was really my boot camp, which got me into this industry and locked me into this industry. Yeah, I wanted to be, but learned a ton there. Again, we had indoor, outdoor, and greenhouse, and so I was traveling all over the place and really started to understand this plant. You know, the company I was with at the time, they allowed me to do a lot of research, which was super exciting. And sadly, sometimes it makes sense not to do a ton of research commercially, but there is that little bit of crossover where money put towards R&D at facilities can equate to money, right? Because whether
Mhm.
it's bringing in different lights which will bring greater yield or whatever it may be, there's
Mhm.
reasons for R&D. But my heart was certainly calling to be able to do that as full time, or at least more than I was getting to. So I decided my time came to an end in California. Right at that same time, federal legalization happened with hemp. And so my mom got one of the first licenses to produce in South Carolina, which at this point in time, as long as you can get a background check, isn't a big deal. But, those first couple of years, they were limiting license, and it was a big deal to get one. So
So is that a
that
research license, or was that 2015, or was that 2018?
18.
Okay.
Yeah, that was 18.
Okay.
So decided to come back. My family and I started the hemp mine, and
Hmm.
the hemp mine is a traditional breeding company.
Hmm.
Selections made for the southeast where it's really, really hot. Have a much different photo period than Colorado and Washington,
Oregon.
where yeah, exactly where hemp was already happening. So there was a lot of work put towards making varieties that would do well in the southeast. and that was successful, but took a while, ongoing process.
Hmm.
So at the hemp mine itself, we sell genetics to farmers, again, mainly in the Southeast, especially in Florida, where photo period's much different. We produce products, so we have our range of tinctures and pre-rolls and that sort of thing. But then we also have the CRC, so the Cannabis Research Center and Coalition.
Hmm.
And this is truly my baby. This is where I do research, and so we do. It's kind of it kind of breaks down into two ways. One, we're a contract research organization, so
Hmm.
we do a lot of research that kind of sucks. I don't get to talk about, but it's perhaps a company that has some IP. They want things done for their internal building of their company and products. So we do a lot of that, but then we also have a lot of open source research that we run with the CRC itself. So the way the CRC works is vendors, so lighting companies, soil companies, they support us to do research. The members vote on that research, and then we put that research out. Um, this is everything from some smoking studies we've done with other researchers, all the way to looking at propagation techniques and putting that research out. The way we get that information out is, of course, through social media as much as possible. But then, we have our login portal for the CRC where we share, everything from white papers to true published research to, just blogs where, we're suggesting, hey, look at this for fire ants in your field. So we try to put out as many videos as possible, and then just about every month we'll have an open forum call where people from around the world jump on, and we talk about a specific product or problem, really, really something that is interesting at the time. So, for example, this month we'll be looking at bulk container packaging for post-harvest, which, on the from a commercial standpoint, has so much more impact than you might think.
Hmm.
Um, and just for example, I do a fair amount of consulting, and when I go into the trim rooms, you'll see people with open containers, big bins with flowers sitting around everywhere. And they say, we've got the perfect drying and curing scenario. And then we bring it out here and it sits in the trim room for 3 days. And, you look at the environmentals and it's 30% humidity in there. And it's like, you have to have some sort
Ruined
of,
it.
you've ruined it. So if you can't control your environment, maybe you can control your packaging. So we do a lot of practical research that, can help the grower that they can take and, hopefully improve their practices with.
I love that because I have so my I did my PhD in cannabis and it started with questions from consumers. So like I'm
Mhm.
very much in the applied sciences mindset. Like I want any research that I do to be valuable to normal people, not just for the science
Exactly.
community. And I feel like there's so much research that's done, especially in the academic setting, that is really just for other scientists in that field.
Right.
Eventually, it might make it out if it's something interesting into the media, but like, you know, it's yeah. So I really like that you're working with companies and actually bringing really good information that's useful. I do remember
Yeah.
I just came across, I was looking up, there was somebody that posted on LinkedIn about ash color, and I was like, I remember somebody posting something about it being related to moisture. And then I looked it up, and I think it was your group that had looked into, because quality has been traditionally associated with ash color. So the lighter the
Right.
ash, the better the quality of the cannabis. The darker the ash, the lower the quality, the harsher the smoke. And it is a really interesting question. Like, what is
It
driving that?
is.
Is it, and people have been asking, is it the nutrients? Is it somebody using synthetic salts? Is it moisture content? Is it pre-harvest, post-harvest? And so I yeah, like but that's so you've got like so many different facets to your research and then like just your introduction there, my brain's going like 1,000 miles a minute because I want to ask you about this and that. So since I just talked about ash color, can you talk about
Yeah.
that? And yeah, just do that because I've got more
Okay.
questions.
Absolutely. Yeah, so ash color has been something everybody's been curious about, including myself. And I've spent so much time reading into tobacco literature and trying to understand a product that's kind of similar but not really similar,
Mhm.
because there's no peer review journals we can lean on to read about it, right? We're here
Right.
in the making of everything. And so, whether it's growing or, you know, uh, extraction, right? People have been doing it for a long time and they've been doing it well. So there's a lot to learn from the practices that are happening. That doesn't necessarily mean it's backed by science. That doesn't necessarily mean that there's understanding in the processes, but there's some understanding to it. And so, like ash color, yes, I believe that you've had very good experiences with wider ash color joints, but that doesn't always correlate with it being a certain practice that we're thinking it should be.
Hmm.
And so, there's so many facets to it. Reading and tobacco literature, right? The amount of chloride that's in the material could change the ash color, because it's burning it differently, and so that produces a different chemistry. There's so many reasons that could change an ash color, and so on one hand it's super exciting that you're kind of starting with a blank page. On the other hand, it's difficult because, as you know, research is expensive, right? And
Hmm.
so if we get funding to do smoking research, we have to narrow it down to something to start with. And so, with the science of smokability, this kind of being a group project with myself and a few other researchers, we had to decide, well, where do we even start? And so, it's easy to keep everything exactly the same, same batch of flour, same grind size, all that the same, and all you're changing is moisture content or water activity. So that was our first phase, looking at ash color. We measured all sorts of other stuff, delivery, how does delivery change? How does burning change? A
Hmm.
lot of things within that study, but one that very easily came out of it was being able to look at ash color. And so ultimately what we found is, um, the the more moist it is, the higher water activity, the lighter the color, which doesn't necessarily correlate with smoking quality. Now, we certainly can't just put the nail in the coffin and be like, that's it of the story. If it's wetter, it's whiter. Like, that's not it, right? There's obviously something else contributing to ash color change, and we just haven't tested it yet. Um, your next study is looking at actual nutrients within cultivation and how that translates to ash color. So, we'll be adding, higher amounts of some nutrients and lower amounts. Well, really, we're going to start with higher amounts of nutrients. So, higher amounts of certain, elements, and um, and compare that to not only a regular fertility program, but then also a flush, and see how that changes it as well. Of course, again, we'll look at delivery of actual smoke and what that looks like, but that's kind of phase two to we've looked at water content as the factor, and now we're moving on to elemental content, which is very complicated because it's not just one element. There's lots of elements in there, and depending
Right.
if you raise one or lower the other, you could be looking at certain things. So, we hope this is just kind of, check mark one, where there'll be plenty more research, beyond our study.
Yeah, no matter what I did and what I feel like it is for everybody, but you answer one or two questions and you come up with six or seven more, and it's like every
Exactly.
I have a pile of Canvas research papers on my shelf, and I would say probably 90% of them, the last sentence is something like more research is needed.
Oh
Like
yeah.
we can't. And something else to look for in the research, and this, is like a lot of the cannabis research right, is done on very small sample sizes, especially things in
Hmm.
greenhouses growing. And I always get the question, like, how come they only have a sample size of 12? How can you make any conclusions with a sample size of 12? And I'm like, well, usually, if you're working in an environment like they're growing a crop, they're not like they don't want to give up the space for the research because they have to like
Yeah.
get rid of it, or they might screw it up or whatever. If it's coming out of an academic setting, it's usually like, oh, the tomato house has given us the back corner to work in, and we've only got, or
Right.
one growth chamber, and we've only can put in this many plants. And so like the limitations, you know, the logistical limitations as well as the financial limitations
Hmm.
are very, very real in cannabis, and it makes it very, very difficult for people who want to research cannabis to come up with a good, solid, robust study with meaningful results that doesn't end with more research is needed, right?
Right. Yep. Exactly. That's so true.
So you said that the lighter ash color had the higher H2O content? Is that what?
Yes.
Okay, so I'm wondering if that is that does that have anything to do with the curing process? Because my understanding is that the curing process sort of decreases the amount of chlorophyll.
Hmm.
Like that's part of the reason that you go through the whole curing process and the reason that you can't rush it. Like if you just put it in a well, it would ruin the terpenes, but you know, put it in a dehydrator or kind of speed up the process, that chlorophyll is going to stay in the plant. You know, it doesn't have time to break down like you want it to in a true cure. So do you think that has anything to do with it? Because if the moisture content is higher, I don't know. Like I feel like
Well, it could, but in this scenario, we cured the flower for the same amount of time, right? It was
Mhm.
just one batch, and then that last 7 days, we took those samples and put them um, in different settings to where they could acclimate to the the different water activities we wanted. Um, so, I guess you could still call that the
So
word
is it
cure
like
is so, it's different for every person. So, technically you could say it was after we dried it, we had them all a matching water activity, and then we separated them out into, their treatments where they could
Mhm.
adjust to reach the water activity treatments we wanted. I guess you could call that 7 days a different curing at a different water activity for those 7 days where it can homogenize. But simply because I've done other experiments looking at chlorophyll, the interesting thing I found, and I'm hoping this can be a paper I get out this year, is that chlorophyll actually 95% of the chlorophyll that's there and present that will degrade degrades within the first 5 days of drying.
okay.
So the amount of chlorophyll that degrades after, it's hit that target dry is very, very minimal, to the point where, I'm trying to give treatment, more extreme treatments to increase chlorophyll degradation. And it's like,
It's already gone.
basically once it's lost a certain amount of water, I guess the enzymes to create that degradation is just not there for it to happen anymore.
So then I have to wonder, is it something that we're not even thinking about or looking at? Because, you know, the whole, and for me, at least my understanding is that a true cure, a good cure, is like at least a month of, you know, you got to dry first, and then you
Hmm.
have to cure, like you put it in the glass jar, and you got to burp them and, you know, leave them open,
Right.
and then, you know, all of the things happen. And if you don't do a true cure, and I'm by no means a master at this. Like I still have a plant hanging upside down that I just forgot to put in a jar, so it's so ruined. I'm not a very good cannabis person, but but yeah, that's my understanding is like it's at least 4 weeks, if not longer, to do a proper cure. But you're saying it's not the chlorophyll based on your research, not
Right.
just with cannabis, but with lots of other things. So is it something in the cannabis that we're not even thinking about? Maybe. I don't know.
I think so. I think so. I think it's something I've yet, or at least anybody I've read or talked to has yet to measure at least. I've looked at chlorophyll, I've looked at other carotenoids and how they degrade, and it's very similar to chlorophyll. I've
Hmm.
looked at bricks, which is an interesting one, but everybody talks about, well, it's sugars. I even used to be a big proponent of that, talking about the understanding of how, sugar starch to sugar degradation, and which makes sense for us to think that because again, in tobacco, that is what they're doing is kind of a caramelizing effect. But they're also drying at really, really high temperatures. So
Hmm.
there literally is a caramelization process going on, but we just don't provide that much heat in cannabis for that to be a process. And I have tested bricks, from start of dry all the way to 60 days after post harvest, and there's no differences. Um, and so it comes down to the fact of yes, through practice, I have seen cure, provide for a better smoke, but what is that? What are you testing to show that,
Yeah.
besides a qualitative test? And so I don't think it's that it doesn't exist. I just think we haven't yet picked the right thing to test.
So the answer is still we don't know.
Yep.
And oh yeah, and so it is okay to say we don't know.
Yeah.
Like and most of the stuff in cannabis, I feel like that's the answer. Same thing with like the indica hybrid thing, like uh what's driving the effects? Like we've looked at THC, we know it's not that. We've looked at CBD, we know it's not that. We've looked at the major terpenes, we know it's not them. And so like
Right.
it's the we keep I feel like we've been as researchers, and it is the easiest thing to do is go for the low hanging fruit, like the most
Hmm.
abundant thing or the most likely answer to test. But
Right.
then I also read papers where they make sweeping statements like there's no such thing. And I'm like, whoa, hang on a second. You haven't looked at
Right.
any, like you've barely scraped the surface of the things that could be the answer. I was really interested in that flushing study. You're currently researching that, correct?
Hmm.
Yeah,
Right.
there was one. Is it? I want to say it's Green RX that put out a white paper, and they're a nutrient company, and
Hmm.
their conclusion is that flushing made no difference. And people are like, "Oh, well, they're a nutrient company." I'm like, "Yeah, but if they wanted to be biased, they would have said, 'Oh, don't flush,'
True.
because right, because they want to sell more nutrients." But it was like
Yeah.
it makes no difference, and I'm like, "So if you want to save money, flush, like pull back on your nutrients. If you
Hmm.
want to just not touch your automated system and just, like leave it alone because it's easier
Exactly.
that way, then do that. Like it's fine.
Yeah.
But the flushing, the idea that flushing also creates a better quality flower still persists. And
It does.
and we still don't know if it's like we still don't know.
Yeah. Yeah. No, I mean, I've seen that paper as well. And then Mike Alden put out a paper recently out of Clemson University looking at flushing, and I think there was another one out of Canada, perhaps. And they all
Oh
show
yeah. It was
very
that guy's
interesting.
masters that he did.
Yeah.
Yeah, okay. Yeah.
Yeah, and all very interesting results. They're showing that yes, you do lower certain elements within the plant material by flushing. Doesn't mess with yield unless you're going beyond 2 weeks. But I mean, you can see on the flower that that there's differences, right? Even if
Mhm.
you're just looking at anthocyanins. Um, so it again comes down to we're just testing, not testing for the right thing to show what
Yeah.
those differences are.
I always find myself trying to think like a plant. Okay, so if I was a plant, the season is the days are getting shorter. I know I'm coming to
Hmm.
the end of my life. I know
Right.
me as a woman, all I want to do is get pregnant and have a baby. I just want to set some seed, right? That's my
Yeah.
whole purpose in life is to make a
Yep.
seed. And if I'm perceiving the end of my life, there's things that I might do to try and increase my chances of catching a pollen grain. Up to
Exactly.
and including, fuck it, I'll make it my own. I'll put out some pollen and I'll fertilize myself, right? So I'm thinking
Yeah.
about these poor female cannabis plants that we keep segregating from their male counterparts, and how are they going to make seeds if we don't let them? Like
Yeah.
that's the purpose of every living thing is to bring their genes through to the next generation.
Right.
Um, and we just aren't letting them do that. And I don't know, I feel like the herming problem is getting worse, but I don't know
Hmm.
if that's because we have increased cultivation and, just communication and all of that. If it's always been like this, or if it really is getting worse as we keep selecting for female and female and female.
Right.
Do you have any insights on my crazy plant thinking?
No, I mean that's how I always talk about the plant too. Even including, kind of explaining photo period. The days are long, you're growing, days are getting shorter, you're rushing to make seed. Same
Or
same
make your
idea.
trichomes bigger so that you can maybe reach out and get something stuck to you to make a seed, right? So you're making
Exactly.
stickier,
Yep.
bigger flowers. Just
Yep.
please give me a seed.
Exactly. Exactly. So I'm trying to think back if my view on if herming has become more. I mean, I went to a consultation recently and they were having problems with herming. My first question always is, well, can you get rid of it? Obviously there's basic cultivation practices we have to make sure, we're not causing like crazy stress, but if it's just one variety, out of everything, well, why even keep it? And of
Hmm.
course the one that herms is always, the fan favorite. So you
Yeah.
have to keep it and you have to figure that out. But I think a lot of people are getting better at trashing the herm, so it's hard to say, are there more herming events happening from seed hunted varieties versus going and getting a variety from somewhere else that's proven more or less? Um, it, I would say it's increasing. This is over my, literally short time span of, 10 years being in cannabis.
Yeah.
Um, so
I
I don't know,
also,
maybe.
yeah, I also think that just like people, plants have different stress tolerances, right? So there
Hmm.
are some strains that no matter what you do to them, they're not going to harm. Or, but there are some that are just like, oh my God, I'm dying. And then they, they're end of life and they'll harm because they're like, I got to make a
Yeah.
seed. And there's everything in between, right? So
Hmm.
there may be, and I think that since we've brought it inside and we coddle it, we give it the best lights, the best nutrients, the perfect amount of water, and we trim it up, and we're so
Hmm.
nice to it that some of these plants have just kind of gotten, for lack of a better word, like weak. They're
Yeah.
not as strong as they would be if they had to survive outside in the wilderness, kind of like a show cat versus a feral, like backyard cat. They're very
Yeah.
different.
Yeah.
Um, and yeah, I'm always making like analogies in my head of cannabis to like things that we can that we know other things about. I'm like, well, it's kind of like this, it's kind of
Right.
like
No,
that.
I know exactly what you mean. I have outdoor cats that, could handle a bear pretty much. And then I've got an indoor flat-faced Persian that is chronically sick and she is treated like a princess. So,
Yep.
it is certainly like that. But it's interesting though. I've got one variety, I've got a couple of varieties that, if you grow it outdoors, right, highest light you can possibly give a plant. Um, Grows fine. No herming, no male flowers. And then if you bring it inside and you're growing it, at 1,000 ppfd, on the higher side, but still, commercially acceptable, um, it will throw herms every single time. Every single time. And it doesn't produce at least viable pollen because the seed isn't made. And then there's some varieties that no matter where you put them, they're gonna herm. So
Hmm.
it's interesting. I'm sure there's a lot of genetics that we need to understand much better to know what's happening. And that's kind of your neck of the woods, right?
Yeah, I guess. Well, so the thing is like I'm a population geneticist. I'm not a, I don't look at genes at
Gotcha.
all, right? So I'm looking at relationships within and among different populations and different
Ah,
groups of organisms. And I was work and the reason I went into population genetics is it's applicable to everything. And I was
mhm.
in on the conservation side looking at rare species and hybridization. I started out looking at cacti and then with a rare cactus in Colorado growing closely to a closely related species, and there was concerns that they were hybridizing because hybrids on the endangered species list don't get covered. So they fall out.
Oh,
They don't cover hybrids. So they were like, okay, so if it's hybridizing, do we keep protecting it where, this is an oil and gas field, Or if it's not hybridizing, can we pick out populations that are, diverse enough or and clean enough to maybe concentrate our resources on? And so that's kind of my background. And we also did some work at that actually ended up being okay, they're not hybridizing that much, and the rare cactus is actually more pure than the common one. And they're actually two different regions that are different species, even though they don't look alike. They're diverged enough and diverse enough to be both protected as more rare, really, because there's two
Huh.
species that were one. And then we were looking at same genus in Utah, two different species. One of the cacti was like really big, angry, hooked spines. And the other one's this cute little, like half a tennis ball, half a golf ball sized little cactus with little short, little straight spines. Just the cutest little thing you've ever seen. Grows in little clumps, like 20 little cactus. Turns out they're
Yeah.
the same species.
Wow. How wild.
Yeah, so you can't use morphology as a good indicator of, different groups or different species. And so, you know, that's where my interest is with using genetic tools is to separate out some of these
Mm.
groups, which could be, hemp and marijuana, which actually do separate out very well, which is interesting. And it's art, and I think it's an artifact of them being selected for different purposes by us
Mm.
for so long. We've created this division and diversity within the two groups. And then
Right.
there's indica sativa hybrid, there's THC, CBD, CBG. Like, do those all separate out into different genetic clusters? Yeah, so that's where my interest lies. Oh, and then, being able to identify, is this Durban poison the same as that Durban poison? You know,
Yeah. Yeah.
which sometimes it is,
Fascinating.
and sometimes it isn't.
Sometimes it isn't. Yep.
But and you working with other crops, like you can have a ton of different kinds of like mums, right?
Hmm.
That are grown for smaller flowers, bigger flowers, different colors, different leaf shapes, all kinds of different stuff. And we've done that to cannabis too. But
Right.
but because it's been illegal for so long, we haven't been able to control it and I don't not regulate, but just keep good records and breed out traits that we
Yeah.
don't want and make things very stable where you get the same thing every time. And that's what you were saying you grew for like big box stores. So like when you go down to Lowe's, all of the petunias look the same, but you have different colors. Like
Exactly.
those were grown in greenhouses. Um, and I mean, that's fascinating too. Uh, yeah, so that's where my genetic interest lies. I'm not a breeder. I'm not a grower. I don't really care that much about genes.
Yeah.
And, and the cannabis plant is so um complex. We've been
Mhm.
messing with it for 10,000 years, and we've got this incredibly complicated genome with all kinds of repetition you, and I think about that and I'm like, nope.
Yeah. Yeah, exactly. I know enough to partially understand, but yeah,
Mhm.
it's certainly not my forte either.
No. So you were talking about the hemp mine and that it's in South Carolina, which I think is so cool.
Hmm.
I want to move to a southern state. I want to go to like South Carolina or Georgia or
Yeah.
Florida, but my boyfriend doesn't want to go there. But so I think it's really cool that your mom got one of the first licenses and your sister, it's like a whole family thing, right?
Hmm. My
And
mom, my sister, my brother.
that's so awesome. And you're growing things for people in the South because we know that if you live in Florida and you buy something from, let's say, Oregon CBD, aka GTR, it's not going to perform the same way it does in Oregon when you move it to Florida. And in
Hmm.
fact, Florida's had a hell of a time finding seeds that will perform and be compliant with the farm bill, which people think the 2018 farm bill, which is fine, but they had a revision of the bill, and then they had the final rules, which went into effect in 2021, which was
Hmm.
total THC. So every licensed hemp farmer across the U.S. has to come in at their pre-harvest test at less than or equal to 0.3% total THC. And you know this, I'm just saying it for the audience. But
Hmm.
but so people in Florida, farmers in Florida, buying seed that performed well in Oregon, where they weren't able to come in and be compliant, because it's a completely different place. Like you were
For
saying, the
sure.
photo period, the weather, the sunshine, everything, moisture, all of that stuff was just completely different, and the plants were just performing completely different. So I've always been interested, and I think this is super important work, and you can speak about this, but we have this idea of seed zones. When you go to Lowe's and you buy a packet of seeds, it tells you what zone these seeds are good to grow in. If you're not in that zone, unless you're a good gardener, you may not be able to grow that plant very well. And I would love to have that information for cannabis so that farmers,
Yeah,
because hemp farming is, it's hard. It's real hard to come in unless you're really dialed in. And lots of hemp farmers have left because they can't do it. It's just, and when you test over, your only option is to destroy your crop. There's no,
yeah,
oh well, we'll just send it out for extraction. It's like, no, you're not compliant. You have to destroy it. And that's devastating for a
it
farmer.
is. It is. It is. You wouldn't believe. Oh, the first 3 years, I would say, was the most devastating. I mean, the amount of farmers I saw come in thinking, this is going to save our family farm, and then they end up, taking a loan out or getting an investor, something like that, and then they lose more than they ever thought they could.
Hmm.
Everything from just selling seed that was straight up marijuana to these farmers to, bringing in varieties that, grow an inch and then flower and you paid $8 a seed for it. Just the disasters that happen was so devastating. And I don't think many people understand to what extent it was devastating for some of these folks. And that's exactly why, coming from the ornamentals where you have trials all over the world, but especially all over the US. If you're a petunia breeder, you take this winning variety, this variety you think is going to be a winner, and before it even is commercialized, there's outdoor indoor trials all over the US where you can experience different climates, different photo periods. um, and then that breeder can license or sell those seeds to the farmers, and they have this catalog for success of how they grow it, how the flower initiation is going to be, just a guide to success for that variety. And here we are in hemp, for example, back in 2018, where it's just like, here's a variety and it's going to be awesome, and that is
Harry.
not how it was. Yeah, yeah. Yeah, exactly. And then, not to mention the problem with potency. There are some states, still to this day, there's some states, South Carolina being an example, our pre-harvest test is 30 days prior to harvest, and it's total THC. There's some that have there's some craziness going on in some states that you can grow things hotter than that, but then there's some that like want finished product to be zero point 0.3, and most of those states really kind of lean towards CBG varieties in order to do that because they're always compliant. But yeah, it's been a fascinating journey, you know, seeing some of the disasters that have happened and trying to do some of the things, similar to traditional ag, to prove success. We took our varieties and we trialed them all up and down the East Coast, with different growers, for free, because that's how it should be. They shouldn't be paying for me to get data for me to sell more things, right?
Right.
So,
Right.
they're trialing these things. Um, and they're getting things for free or because they're doing that, and then that way the next year we can all be more successful with saying yes, this had too short of a flower initiation time, or whatever the case may be. So we spent a lot of money and years trialing in different regions, and that taught us so much. I mean, the varieties I can sit down and say, grow these two in South Florida, but if you grow those two that I said you should go in South Florida, up in Michigan, it's going to freeze before they're finished flowering. So, so many resources that we're still lacking. And once cannabis or federally legal cannabis is happening, we're going to have that same ordeal with growing outside at least.
Hmm.
It's even more complicated. You don't have a timer. You can just say on off. You can't empty your room because it's hot and humid. you got to deal with what nature gives you.
Yep. Yep. In New Jersey, we have a 15-day harvest window, which makes it even harder.
Oh wow, yeah,
Yeah. And so I had to monitor our flower pretty closely. And as soon as it hit 8%, we had to call up the state and be like, we need to test. And they'd come
yep,
out. And we hit it every time. We were never not compliant. But like I had a schedule, like, and we
yeah,
could not screw if we screwed up that schedule, it screwed up everything. I mean, they're still growing, I think, but I don't know. And they brought me in to figure out like what can we grow here in New Jersey with the 15-day
hmm
window and still be compliant in hoop houses where we don't have a
right,
whole lot of control over the everything.
yep,
And what can we do to, things like what can we do to decrease costs to, keep the lights on for as little time as possible, depending on the season, because they grew year round.
hmm
Um, and just, what, like, how ca can we adjust things with the nutrient schedules? And it was aquaponics too, which throws a whole nother, like, thing
Woo,
at,
yeah, yowzers.
yeah,
So that's a lot of moving parts.
it was kind of like learning to drive on a really shitty car where,
hmm It's
only
icy
one
out.
one windshield wiper works and the brakes don't really work, so you have to also use the emergency brake and first gear doesn't work, so you have to start in second. Like
Sounds
very,
rough.
very fun. Well, I learned a lot though,
I bet, yeah.
so, um, and I learned how to write SOPs and how to come up with IPM, whatever schedules. So I learned a lot, but haven't been doing that for a while. I do miss growing. Do you get to go out and do a lot of growing, or are you mostly running around helping
I do a
people
lot
set
of
up
running
hives?
around, but I like right behind this wall is our grow chamber. So every day I take a few laps, just see everything. And so I still have a fair amount of time in the garden, but not as much as I wish there was more time in the day, right? I'd
Yep.
spend a couple hours out there, just trimming. I mean, even just doing leaf removal, the things you can see with a plant and, develop an eye for, whether it's pest control or noticing early fungal problems or, noticing different flower initiation times. Yeah, the longer you get to sit with them, the more they tell you.
Yep. True that. And hand
And it's
watering.
therapeutic.
And hand watering will make you do that.
Yeah, for sure.
But hand watering, 4 out of 10, somewhat recommend. But
Yeah,
really, if
yeah,
you can automate your watering, it is much better. But yeah, hand watering, you get your eyes on every single plant, which makes it,
that's true.
yeah. So, what other research are you doing? Because I know I have a feeling you're not just researching plants, you're also kind of teaming up with folks who are looking at consumers. Is that, or am I off base with that?
Guess the most consumer driven is the SOS study.
Science of smokability,
Yeah,
right?
science of smokability. Yep.
Okay, so
Yep.
and that's is that still going strong? Because I know they were doing some really good research,
Yep.
and you're all still a team, and
Yeah, yeah. So the reason it might seem like things have dropped off is because in our first study, where we were looking at water activity, the end of the study is smoking, right? And we smoke on a robot. So it's this a smoking machine
Mhm.
put out by combustion. And so, the machine is hooked to a computer, and in the computer, you have to put in a topography. So, tell it how long to pull, how hard to pull, the duration. And so, it ends up looking like a heartbeat of how you should have it smoke your joint. Um, but the problem was, as we're doing this study, the recipe, the topography we had to put into this machine was Health Canada's cigarette smoking topography because that's all that exists. And so as we're watching this happen, we're like, this is really, really wrong.
It's not how
Um,
people smoke
this is not
joints.
how people smoke. So, we finished the first study, but the next decision was, well, do we continue to do this work, knowing, just visually it's wrong. I'm sure on the other end there's going to be differences. Do we fix it? How in the world do you fix it? Or do we just keep rolling? And so we said, we're going to take time and figure this out. And so that's what we did. Um, the company Cam Combustion, they actually made us a flow meter to where we could hook up a joint to a little device. And, it's small, it's a little clunky, but people at conferences and different events we went to, we had actually smoke a joint. And so we had 50 or so people smoke a joint, and we were able to, statistically pull out the different types of smokers. And so there were three types of smokers. Of course, it's never just one answer. But there were three types of smokers. There was the intense puffer, so one who, you know, just pulls right away, really hard, really fast.
Mm.
There's the slower, less intense puller. So, it's ones that takes a little more time, I guess, see how it feels on your throat a little bit. And, the pull is longer, but it's less pressure. And then, of course, there's the third, most unique smoker that's a double puffer. And so those who, hit it medium puffs back to back.
Hmm.
So statistically, the most was the intense smoker. And so now, literally last week, we were going through our final edits of that paper. And that way, other people who are doing smoking studies can take that topography, put it into their machine and have something that truly represents cannabis. But that took a good year to do between, getting enough samples of people smoking and putting all that into analysis and figuring it out. So 3 weeks, we will start doing our next round of tests with our new topography. So that's why it kind of like, we had all this out there about our first study, and it kind of died off a little bit. And that's why it's because we had to get our machine right.
Well, that's super interesting, and also speaks to the reason why research takes so long.
Exactly. Exactly.
Uh,
Exactly.
and so did you go to conferences to find people, or did you like recruit people from the internet, or like how did you recruit
Yeah,
your
so I
participants?
I, I packed it up and took my little briefcase to a number of different conferences, some cannabis events. Being in South Carolina, I didn't, I wanted the smoker to be experienced smoker, right? I could go to the smoke shop down the street here, but I wanted people in the industry that are confident in the way they smoke.
Hmm.
And so, we try to sample experienced smokers at conferences and special cannabis events.
Did they know who you were?
Yeah, they knew what they were signing up for.
That's so cool. So what else is going on? What else did I
Well,
write down here?
let's see. Um, one very interesting thing that's kind of just been a union is working with American Air Filters. And so they're a company who makes filters for HVAC equipment, for actually all sorts of stuff. But I think the normal person would know them from just the square filters you put into your HVAC. And with them and their teams of scientists, they're coming from a point of view of their cannabis is just getting to where it's Schedule 3, just getting legal. We have a world-renowned, multinational company. We got to be careful how we get into an illegal
Hmm.
industry.
Hmm.
And so, right now they're at the point where they want to help the cannabis industry. They want to get into the cannabis industry, but they also want to, um, before they jump in saying they know everything, they want to understand what's going on in indoor cultivation. And so I've traveled around to a few places with their science team, and we've looked at filters, we've looked at different facilities and how their airflow works, and going back to the few people that have passed due to air filtration problems ultimately, whether that was masks or internal, I don't know, but it is particulate in the air that's causing the problems. And so working with them, a company who has put so much time and research to, particularly the swine industry, an industry where if virus gets through, one house to the other, you've got dead and mummified piglets. And so, there are people who have figured these sorts of things out. So it's been awesome kind of getting to understand their knowledge and their standpoints of other industries and wanting to see how they can help cannabis growers do the same thing. So they've got a really, really cool facility. Um, on the East Coast. And so, cannabis growers have been sending in, used filtration, used filters from their HVAC. And, this EAF is, looking on it with a scanning electron microscope to see, well, what's really happening if your filtration intakes 50 feet in the air. Is it insects on there? Is it dust? Is it trichome heads? Like, what's in these filters, and how do we need to approach that? Because to lack of my knowledge, filters are very complicated. There's all types. There's types that you should use where there's high humidity, because if you use a certain type where there's high humidity, well, particulate gets stuck on there. It easily allows the moisture to stay, and you're just growing things on your filter. So, they're coming from a standpoint of, let's look at all these pieces and make some really wise decisions versus just going down to Lowe's and buying a random cheap filter. So that's something that, has just kind of begun the past 6 months, and something that's been absolutely fascinating. So, you know, over this past 6 months, they've been collecting filters and doing samples, and we're hoping to put perhaps a white paper. I'm not sure how they want to publish quite yet, but we're going to put something out to kind of give growers feedback of, hey, this is what's going on in these anonymous grows, right? We're not trying to call anybody out. In your anonymous grows, where do you line up beside this? How do you think about your filtration, and what are you doing? So, hopefully, at least in those areas, we can, make things healthier, less pathogen spread, less disease spread, and hopefully a happier place for people to work in because they can breathe better.
So we're thinking about this in terms of both human health, so the workers that are in the facilities that are making contact with the plant and working around the plant, not just in the grow houses, but also like the trim rooms are a big deal
Exactly.
because they're
Hmm.
using dried flower. There's potentially mold spores on the flower if the flower's not clean. There's all kinds of
Right.
particulates flying around, but also from a plant health perspective, right? So
Hmm.
in the grow
Absolutely.
facilities, PM is a really big problem, powdery mildew.
Right.
Um, I don't even know what you do once you get it, really. I mean, everybody has a problem with it at some point. It doesn't make mycotoxins, but it's also not, I mean, it's still mold, right?
Right.
So, and it's, correct me if I'm wrong, but it's usually due to a moisture issue. But once it's
Hmm.
in a facility, it's kind of hard to get rid of, right?
It is. It is. It is hard. It is hard. It's easier to be preventative and keep it out. There are ways, but yeah, it's hard. It's hard, and once it gets bad, it becomes a snowstorm, and it gets really bad, and it takes a while to work that out of the system. In cannabis, for good reason, we've got a lot of restriction on pesticides we can use. So, depending on what state you're in, some states you can use elemental sulfur, some states you can't. Both of those are, considered organic, not even a pesticide per se, but that is a good one for powdery mildew. But yeah, it's tricky, but exactly, it's not just health of the people, which obviously is the most important, but it's also health of the plant and how to not keep reintroducing problems that you have.
Right. I know that I've got plants in my yard, not cannabis plants, but like other plants that have health issues. Like
Yeah.
my hydrangeas always get powdery mildew. And if
Yep.
I was
Mine
to grow
too.
cannabis in my yard, I guarantee you that my cannabis plants would get it from some of the other plants that are in my yard.
Yeah.
And I can never get rid of it. I don't try terribly hard, but I'm
But
not a
it's
very
there. Yep.
I'm not an avid gardener, so but there, so powdery mildew is an interesting one, and like I said, pretty much everybody's going to get it at some point because you can bring in the spores like just from your own garden. So,
Right.
if you're not suiting your employees up, you one of them could bring it in on their shoes, on their clothes. So, and I feel like you can be as careful can be, but some of these pathogens dirty little buggers will get in any way they can, through
Yeah.
insects, through, us as vectors, bringing in clones from another facility that are infected with
Hmm.
something. There's even some pathogens that are spread through seed. I think
Right.
the latest data on hot plate and viroid is if your mother is infected, it's transferred to about 10% of the seed. I don't know if that's accurate anymore, but
Mm.
that was the last that I heard. And you working closely with cultivators, I've been interested because I feel like I've been looking. I mean, I've been looking for the research on this. It's more speculation and companies making um, guesses, really, about how devastating some of these diseases can be. And I want to talk about hot plate and thyroid just because it's not an obvious disease. Like it doesn't
Hmm.
outwardly show a lot of symptoms. Um, Unless you've got a serious and then you get smaller plants, the flowers kind of fail, your yields go down, and it's not just your
Right.
yields, right? It can be quite a large chunk of your yield, but also your
Yeah.
trichomes suffer. So your THC can drop as much as like 30 to 70%, 30 to 50%. Uh,
Yeah.
Total THC, not like 50% THC, but
Yeah.
so if you had like THC that was testing at 30%, you get hot latent thyroid, it could drop as low as 15%, which then drops you down into a different price category for what you can
Exactly.
sell that flower at. So have you done any research or have you seen like what's your feel for how devastating HLV can be? Because I feel like there's also some cultivators out there that are just like, well, it's not harmful to us, so like why? And we don't have to test for it, so why do
Yeah.
I need to test for it? And I'm like, well, because of your pocketbook, that's gonna
Yeah.
kill your bottom line. So can you
Yeah.
talk
No,
about
it's
these things? Because I feel like there's no numbers out. There's no like, I haven't found any research out there that with hard numbers of these cultivators who have had this pathogen have lost this much money.
yes. Yes. Okay. So I don't know if I'm gonna have. I don't do a lot of work with HLVd.
Hmm.
Not specific research. All right. I've dealt
Right.
with working with growers who have it. When I was in California, we had it. We had it before we even knew what it.
What it was.
That it had before it was even identified. We just called
Mhm.
them duds, um,
God. Yep.
which they were certainly duds. So I've seen myself the loss that it can bring a facility for us, just to give you a very specific example, we had about 10 varieties we would run, and the only one visually it would affect was grape pie. And of course, that was everyone's favorite, so we couldn't just get rid of it. So, we're sticking, you know, 50% extra, transplanting 50% extra just to kind of make up for that. So, and in each room, marketing would demand, a third of it be planted in grape pie, or sales would demand that. So, for each room, if we're growing a third in each room, I could always guarantee at least a quarter having 50% less yield. That equates to a lot of money over time, a whole lot of
Mhm.
money over time. And at the time, had I had testing capabilities where, I can make, my mother stock area, you know, clean, roped off from other varieties, where I'm continually testing the leaves and the stem and the roots because, it can show in different places. I feel like I could have significantly made my company more money had I had some exclusion type protocols like that. But at the time, like I said, it wasn't even identified yet. Working with companies now, they do invest a fair amount of money for testing, but again, if I'm losing 25% of my yield because I'm having to grow these varieties that, are more susceptible of it, that equates to a lot. So being able to have clean mother stock, at least that you're pulling from down the line, can save a lot of money. But it is frustrating, and visiting other facilities as well, I wouldn't just say a lot of the industry's failure in general is only from HLVd. I think that's a big one. But like in my example, it didn't even affect the rest of the varieties I was growing. It was just this one.
Mhm.
Um, but what I do not necessarily see enough people talking about is Fusarium.
Hmm.
Um, you know, almost every grower I've ever been to that has tested, not everybody wants to necessarily know what they have, but that has tested has Fusarium. And there's skills within cultivation that can help you. Um, you're never going to eradicate it, but you can at least keep the pressure down enough to where it's not affecting yield. But there's a lot of growers out there that, you know, they're overwatering, and those problems just, compound on top of each other. And I've seen more yield loss in that than HLVd. Again, in my experience, I'm sure there's other scenarios and that other people have experienced, but in my experience, Fusarium is certainly another one that is just everywhere. And the problem is
Fusarium is,
it's coming from a lot of the media that we're growing the plants in. So
so the Fusarium is it a fungus?
it is.
And it kind of it's like root rot, right? It causes root
It
rot in
it
some other
does,
plants?
but it's so gnarly depending on the strain you're working with. It could affect, give a stem canker. It could, probably 50% of the people that think they have botrytis is actually Fusarium,
Interesting. So it
bud
causes
rot.
bud rot as well.
Causes bud rot, causes, it'll kill the roots too. So it's all sorts of problems that it can cause. And when it kills, it kills, right? It doesn't just stunt it for the most part. Um, so yeah, those two are probably my most feared for growers with having to deal with and fusarium stuff because it comes in on the peat and it comes in on the cocoa. Um,
Oh,
so
interesting.
you can spray all the microbes you want, but you're just bringing it in as well, which leaves you with a very unsustainable option, to completely avoid it.
So what do growers do to avoid, like if so I always say like test early and test often, especially for something like hot blatant thyroid. If you've got, people are doing tissue culture now where
Hmm.
they've got backup stock in tissue culture so they can switch out their mothers like every,
Yeah.
3
Oh yeah.
cycles or something like that so that they can make sure that the mother's clean because if you lose your mother,
Right.
yeah, you're in trouble.
Hmm.
But you also don't want to have everything in tissue culture because that gets really expensive really fast too.
Exactly.
So is there anything you can do if you catch fusarium or botrytis early? Or is it like if you catch it because you're testing early and testing often, if you get a ping for fusarium, are you just fusariumed? Yes.
Yeah. Yeah, for the most part. I mean, the only way to truly get rid of it is it's everywhere. It's ubiquitous, right? It's all over outside, just like PM is. So it's hard to never get it. So, all you can do is,
Cull
of,
it.
of, of, of course, you can call it. You can go through, having tissue culture, having clean mother stock. Again, being very, very careful about where you're getting your growing media. And if you truly want to have 0% chance of not getting it from the growing media is Rockwool. That's not my favorite option because it's so unsustainable and it's harder for a grower to use, right? There's less buffering capacity there, larger failures if you don't have a really great irrigation system. Um, but testing often, culling really bad ones, but really reducing stresses because, I've seen grows where they have the problem, they go through veg, they go through early flower, you know, into later into flowers where you'll really see overwatering problems show up. And that's where, they're overwatering, the plants are drinking less, and maybe their timers aren't adjusting for that. And so then fusarium kicks in, it takes over, and then you'll see, whole rooms fail.
Mhm.
So, if you're gonna grow with active fusarium, you've got to be a good grower that are not providing a lot of stressors for the plant. But I would say it's in most rooms. You might not see it unless, you have really hot event that stresses the plant, or like I said, a really, really gnarly dry back. And then that disease
Stress is
kind
a
of takes over.
So stress is
Yeah.
a big trigger for it. So
Oh yeah.
it's probably around, but as soon as you get a big stress event, then the chances of an outbreak like is kind of like when you get sick during finals week.
Yeah, exactly. Your immune system's down and just gets you.
So
Yeah.
I was talking to Michael Deleg, who works with microbes, and he had a really nice analogy about microbes and pathogens. He thinks about it like a dining room table, and there's only so many seats at the table. And if you can fill those seats with good microbes,
Mhm.
there's less seats for the bad ones to come in and take over. If you are in a totally sterile environment, then the bad microbes can just pull up all the seats and fuck everything up. And I thought that was
Yeah,
a really good analogy because, some people don't really, I don't want to say they don't grasp the concept, but they don't utilize or don't, like, there's tools that you can use, like adding, different microbial concoctions to
sure.
balance out these whole ecosystems that
Mhm.
work together outside of what we can see.
Yeah, yeah, exactly, exactly. That's a good way to look at it. There are certain, the way I look for at beneficial microbes, so like a trichoderma or some sort of drench that you would put in the pot to increase your microbial load to void off the pathogenic microbials. But actually, a lot of my grad work was with this, looking at different microbial, biological microbials and how they can interact with Phytophthora. Um, and what I found in the study, and kind of what I've seen to this day, is those are great. And if you have a light pathogen load, they'll help fight that off, right? They're taking up a lot of those seats at the table. But if you stress the plant's immune system enough, they're not going to be the savior for it.
The number of
Um,
seats doesn't matter anymore.
it doesn't matter anymore. So, they're great as an immune booster, but there comes a point to where the plant's so damaged that, the microbes, the pathogenic microbes just kick right in and take over. So, they're great to have there as preventative and to help and to kind of give a little bit buffer. If your stress is, if it's 100% kills it, if it's at 30% and you have microbes, it might bounce back. But if you go up to 60% with your stress, the microbes aren't going to do much. Um, is kind of how I look at it.
So life is such a
Yeah.
balancing act.
Right. It's
Uh,
like you or I. If your immune system's compromised and you get COVID, you might have a harder time than a healthy person.
or you might
Um,
die.
or you might die. Yeah, exactly.
Which sucks. Um, so that's interesting. So what exactly, like, obviously we both went through PhD school. Um, what exactly was your PhD? Like, I mean, and PhDs are super intense and you have more than one study and you're doing more than one thing and everybody's heard about mine, but what exactly did you do in PhD school?
I love how you say that, PhD school. Well, my PhD school, it was under Dr. Jim Faust, who is a floriculturalist. And so, because of that, my host plant was poinsettia, geranium, and mum, petunia, floriculture crops. Greenhouse production always, but I was super interested in microbes, right? I thought
Mhm.
biologicals were the bee's knees, and fungus was still exciting to me, everything from mushrooms to, pathogenic. So I was very fortunate, and he kind of let me pick my project. And so I did, a year's worth of while taking classes, I was trying to figure out what I wanted my project to be. And at the time, there was a lot of studies coming out of India where they had found this mycorrhizal type fungus. It wasn't technically a mycorrhizae, was it technically a mycorrhizae? It was an endophyte of sorts that acted
Mhm.
as a mycorrhizae. Minor differences, but in that world, those differences are very important. But it was like a mycorrhizae. And this mycorrhizae was found in the desert, which, where a lot of mycorrhizas are found, right? In places where the plants are going to struggle to live. And so having a little bit of boost from another organism to create this happy symbiotic system is beneficial. So it was found in the desert in India. and they were doing all sorts of studies, showing it helps take up water, this and that. And so I said, this is awesome. I want to work with this fungus. I want to see how I can use this fungus in commercial production systems and save everybody fertilizer and water and disease, and
Hmm.
save the world with this fungus. So it took me about a year, which I didn't understand the process at the time, right? I was just very young and excited and willing to do whatever. And, it was about a year for the USDA to finally allow me to bring this fungus in because it wasn't something that was naturally here and on our land. And so makes sense, you don't want to
Yep.
bring something over that just takes over the world. So I brought it in, and so I was very restricted on how and where I could apply it. But ultimately, I cultivated it myself and applied it. And so within my studies, I took my host plants and I said, well, with this fungus, what can it do on a commercial system? I looked at rooting. So this fungus and other studies had shown to increase auxin production. So I said, all right, sweet. Well, maybe I can use this in propagation. It increased auxin, it increased adventitious root formation, and perhaps we'll have a natural alternative to PGR, rooting hormones. So that was one of my chapters. Another chapter was looking at nutrient uptake. Well, let me go back to the propagation study. It didn't significantly increase rooting. That was too bad.
Uh-oh.
Yep, right. You're gonna have a lot of those. Well, didn't show anything, but I guess that is showing nothing is also showing something.
Yes.
So that was that chapter. With nutrient increase, I was really looking at phosphorus because a lot of mycorrhizae in general, there's only a few nutrients that are actually helping the plant to uptake and increase. And so phosphorus was one of those. So I withheld phosphorus. I had phosphorus at regular levels, and then, I applied this fungus, and then there were the controls. What I found was if you are applying everything the plant needs from a fertility standpoint, the fungus does not help. If you're lacking things, then the fungus helps. And so, coming from an industry where, you know, saving a cent per thousand pots is not important. They'd rather just, what I mean is like withholding phosphorus where this fungus can then take it up. It didn't make sense, right? It didn't make sense for them to take out the phosphorus and just apply this fungus when they could just apply the phosphorus themselves.
Right.
So, to my disappointment, that did not work out. At least, I mean, it was very interesting and scientific, and I learned a ton. And we learned a lot about this organism. But from a commercial standpoint, it was like, well, that's nothing that can be implemented to actually help a grower. Um, and then the last chapter was looking at its effect on Phytophthora, which is another fungal pathogen. It does occur in cannabis, but not quite as much as certainly as Fusarium. So
Mhm.
I got to learn how to, grow this pathogen, inoculate plants with this pathogen, and then of course I would have my treatments with the fungus. And what I learned very quickly is, and also in that treatment, I threw in other commercially available microbes, so like root shield and some of those, just to kind of see how my fungus compared to the rest of them. And really, with all of the products, if you have an infection that great, which was me cutting the plant and, actually inoculating it, microbial is just not going to do anything.
Mhm.
It's not going to win. But, if it were a natural environment where it's randomly getting one spore, and then the plant's covered in these microbes, perhaps it would help. But from an inoculation of inoculating a pathogen on these plants, it just wasn't enough to withstand the pathogen, at least
Mhm.
with Phytophthora. So,
Uh, Interesting. I live in New Jersey, not too far from Rutgers, where
mhm,
Dr. James White is, and he does a lot of work on endophytes. And he had some really interesting ideas, and he had a grad student, and I don't know if she's graduated yet or not. Her name's April, and I can't remember her last name right now, but she was really interested. So they have a hemp program at Rutgers, but the cultivation department doesn't want to work with the mycology department. And so they would never
Oh,
give her space to try to experiment with endophytes, which is really uncool. So
yeah.
so we were going to give her space to do experiments at our farm. Like we were going to have like an experimental area where she could come and inoculate various, plants. We were growing all the same strains, so it didn't matter, but inoculate with different endophytes to see if any of these endophytes have the potential or the capacity to like how to like trigger the plant to
Hmm.
up regulate THC production or cannabinoid, like something as
Yeah.
a response to being infected with something that's not going to like kill them, but they're like, because aren't there a lot of plants where their response is to do something
Hmm.
not necessarily bad, but like they do something and for us it might be adventitious depending on how what we're growing
Yeah,
the plant for. So
absolutely.
are you interested in doing stuff like that with your research at all, or are you
Yeah,
going,
absolutely. No,
yeah.
I haven't got to do a lot in the past couple of years, just because that's how the journeys took me. But yeah, no, I was very lucky in grad school. One of my advisors on my committee was a mycologist there at the university, and so she, held my hand and walked me through how to grow all these things. I took her mycology course too, of course. But she let me use their lab space to grow all my little critters, and so that was fantastic. But no, I would absolutely love to get back into that sort of thing.
Well, I should connect you to either James if you don't already know him. He's a great guy, super smart, super interesting. His research is super interesting. And I like I said, I don't know if April's still there, but because you have the capacity, the space and the yeah, the
Yeah,
know-how
no,
and
I
knowledge.
love collaborating, with universities
Yeah.
and grad students. Yeah, it's right up my alley.
Well, I'll connect you. But I feel like, and it might be the case where, you inoculate and then it doesn't make a statistic, it does increase THC, but it's not statistically worth it to
Right.
go
That's a lot
around.
of studies.
Yeah.
A lot of studies.
Yeah, I feel like that might be the case. But wouldn't it be interesting if you could
It would
increase?
be fun to try.
I was really interested in epigenetics. And,
Hmm.
when plants are stressed, they respond with, upregulating, downregulating. And sometimes it's just a Hail Mary, like, fuck, I just need to make it through to the next generation.
Yeah.
And so plants will pass on some of those epigenetic marks to their offspring, to their seed offspring, so that that offspring has a better chance to survive in that environment that they're going to get dropped into because
Right.
can't move, right? So
Hmm.
the offspring has to be able to survive, and if that plant can somehow make it through, then those epigenetics will get passed on to the next generation. It's not the same in animals because I mean, we're just scratching the surface with epigenetics, but I had a theory that, and I was talking to Max Jones from the University of Guelph about what is the potential for, epigenetic changes to occur from cloning stress, not only
Hmm.
to, the clones that have been cut, but the mother as well. Like she's getting
Yeah.
cut. Like that's got to be stressful, getting bits and pieces cut off of you, every few months. And then the clone, the poor little baby clone is now it's an own individual. It has to create roots and shit, things it's never done before. And I'm like, that has, again, thinking like a plant, right?
Yeah.
That's got to be super stressful. So I grew some plants and I was doing like clones and clones of clones and clones of clones of clones and testing
Hmm.
both the mother before cutting and after cutting. And all I wanted to look at was like the methylation sites, which are, a mark of an epigenetic change, and
Mm.
never actually finished it. But I'm thinking if you could somehow master epigenetic manipulation, you could make the plants do things like um, flower earlier or
Right.
pollinate later. There's all kinds of things you could do, which, yeah, when I'm
Can I
not
tell you
in a,
about this talking about, clones and taking clones from my mom for a long time, yada yada. I've never had my mind set on, how. there's people out there that claim, if you clone off a mother 3 times, it's going to completely change its genetics, right? There's all sorts of stories of how this works, right? And I've just kind of sat back and been like, I've cloned some plants for years and they're
Mhm.
fine. And then all of a sudden, this past year, I have this one variety that I've been cloning again for years. Um, and it started shooting off a little bit of variegation, right? And I've had it tested. The test, it's only going to show a positive for what was tested for
Mhm.
as far as virus goes. Had it
Mhm.
tested for virus, nothing shows up. It looks just like a sport to me, right? Like a natural variegation,
Mhm.
not necessarily viral. Um, so I love variegation. I love
Me
variegated
too.
plants. I'm
Me
from floriculture, right? Like every
too.
house, every plant in my house is variegated. But so I see it, and after I see that it doesn't necessarily, I know I can't say this with 100%, it doesn't look like virus, but when I thought that, I was excited. Um, a couple more months went by, and this plant started putting out shoots that are only the color of the light part of the variegation.
Mhm.
And I've had plants do that before where it's just here and there, like it'll be a little bit and then it'll die off because it doesn't have a lot of chlorophyll. It's wild because with this plant, there's like enough chlorophyll to where those branches will become full. And so
Mhm.
it's not all the way white in the white part of the variegation. It's like a darker yellow, but still
Mhm.
significantly different color than the normal green. But I
Mhm.
just never seen a plant like it. It is so beautiful, but from a mutation genetic standpoint, something super funky's going on. I don't know how to explore that anymore myself, but I would love to for somebody to look at that because it is just wild.
And my brain tells me that that could be a couple different things. And, I've had people tell me that variegation only comes from viruses, and I think that's absolutely not correct because
I do too.
it can be a mutation. Like you can have a mutation that causes lack of color. Like
Right.
it happens in people, it happens in all kinds of animals and plant. Like it doesn't make any sense that it would only be from a viral or a pathogen infection. And I've personally had a couple of different variegated cannabis plants. I grew one called White on White, which
Hmm.
was it was all over the plant, not white and green, like not that but it was very like speckly everywhere, the whole plant.
Hmm. Okay.
And the guy who gave me the clone said, it could be hop latent viroid. But this was like back in 2018 before they even knew that hop latent viroid was in cannabis plants. And it was fine. It didn't show any signs of, sickness or
Yeah.
anything like that. So I'm like, I don't think it is a latent viroid. I think it's just the color of this plant. And
Yeah.
then my friend grew a chemdog plant that was very variegated, very yellow in the light
Hmm.
parts and very normal green color in the green parts. And he took a ton of
Right.
pictures for me. And it was chemdog. And I've talked to people and they're like, oh yeah, chemdog's naturally variegated all the time. Like
Oh,
it just is. I'm like, okay.
fascinating. Yeah.
And I need to talk to the guy who is actually chemdog. He surfaced on my Instagram, and I'm like, I gotta have this guy on my show to talk
Yeah,
to
that'd
him
be
about
cool.
how the legend of chemdog, which I mentioned in my PhD work because I traveled around to all kinds of dispensaries in 3 different states, and I didn't know anything about cannabis back then. I didn't know anything about strains. I just kind of bought what I thought was popular at the time,
Hmm.
and different dispensaries. There was Chem 91 and Chemdog with a DOG and Chemdog with a DAWG and Chemdog 4. And I was like, Chemdog D. And I was like, I don't know what all of these are, but if they had like some sort of variation of Chemdog, I was like, all right, I'll buy that. Because I thought they were like maybe local variants or something like that up, but I was like, oh, they're probably related, I don't know. And then I looked up what is chemdog, and it was like this whole thing about this guy who found 12 bag seeds, and the first year he grew four and he called them 1, 2, 3, and 4, and one made it through it. And so chemdog 1 eventually got changed to chemdog
Oh.
91 because that was the year that he grew it. And then the next time
Mhm.
he grew four seeds and he called them A, B, C, and D. The one that made it through was chemdog D. Like, and I was like,
Fascinating.
oh, okay, that makes sense. And it was really funny because genetically they were all like as similar as you would expect from a sibling relationship. So
Hmm.
that told me that, yeah, all these are different seeds from the same
Right.
parents, which was really, really cool.
That's cool.
Yeah.
Yeah, that'd be fascinating to get info from him of how all that went down.
Yeah, yeah, because I have it like laid out in my dissertation, but I was like, I don't even know if this is, I don't know if it's a legend, I don't know if it's real, I don't know if it's true, I don't know if it's accurate. So I would really like to know.
Yeah, cool. That's
Yeah.
super cool.
Well, so what else do you got going on? Anything else that you're excited about or any
Yeah,
research coming
I
out?
do. I do. One of my other kind of, top three projects I have right now is with Pip Horticulture. They're the racking company. You know what
Okay.
I'm talking about? If you've seen pictures of indoor grow rooms where
Yeah.
it's white racks where they're stacked and they
Mm-hmm.
roll and they'll move.
Hmm.
That's Pip Horticulture.
Okay.
And so, for them, funny story with them, actually, it goes way back to being in San Diego.
Mm-hmm.
Um, when I was in San Diego, my first research project, significant besides just internal R&D, was looking at light intensity and how that affects yield with
Mm-hmm.
Fluence horticulture, back in 2016.
Hmm.
And Pip was, they've been a company that's been around for a long, long time, but they're in like the back of stores moving, packages, for hard goods, not moving water from one level to the other. So my start with them was actually being, I think, their number two install back in 2016, where I was being the guinea pig for, how we move the water, how we hang lights from these things.
Mm-hmm.
And then that led to the Fluent study where we looked at lighting. Anyway, fast forward, our relationship came back around where they said, "Hey, we move air in our systems, right? They have fans they sell that run air from, horizontally top to bottom. But they're like, there's no research out there that says how much air we should apply and how this should be done, right? We're growing good systems, but we really want to be able to say an intensity to give for how much velocity, how much velocity should be given to these plants for air movement." And so we started, at this point, I think we're in year two, and it'll last to 3 years looking at airflow. So it's been a lot of research gone into this. Um, and, of course, I start my journey by digging into literature with other crops, and there's just nothing for cannabis, right? You've got growers that have giant fans, that have oscillating fans on the sides of there's all sorts of scenarios.
Mm-hmm.
Um, and so we said, all right, well, we're gonna dial this down a little bit. So we started with, having three identical rooms, all having different airflow. There was definitely a few trials in the beginning to narrow it down to these levels, but we're at 200 and 400 feet per minute. So, just for those who don't necessarily understand what those numbers mean, zero is bad, right? There's no airflow. High would be pretty aggressive to the point where if you went up just a little bit more, you'd probably be getting wind burn and nutrient, tip burn on your plants because there's so much wind. And 200 is about that mid-range zone. So, at this point, we've gone through about 3 reps of growing at these different air intensities, and it's been absolutely fascinating to do this research and, thinking back a couple of years ago of how I thought about air velocity and air movement within an indoor grow and what I know about it now. When I first went into it, I'm sure you learned in plant science classes, when trees get more movement, more velocity, right? They strengthen, and so they actually become shorter, because they don't have to grow it as tall. Well, that was my walking theory because, you know, why wouldn't it be? Um, and so after, adequate reps with this, we have seen that adding air velocity actually makes the plants taller, like significantly taller, 3+ inches, depending on which treatments you're comparing. Um, and not only is it making it taller, it's allowing the plants to transpire more, which then takes up the nutrients, gets those nutrients into the plant. higher elemental content. We're tracking that weekly within the plant from the higher airflow. Um, and ultimately, it's equating to significantly greater yield. Significantly greater yield. And so, looking back on this research and thinking about it, it makes perfect sense. You really should think about a grow room air movement in two ways. One is your macroclimate, right? You're homogenizing air just to make sure, what's coming out of your DHU and your HVAC system is making the room itself a homogeneous level.
Mhm.
And then you've got your micro, where you're breaking boundary layers, right? You're actually moving the air off of that leaf to where the stomata's opening and it's breathing more, right? It's circulating that air. CO2 is coming in, CO2 is coming out. It's breaking what's around that leaf layer. The plants are drinking significantly more, obviously, but and so, from the plant physiology side, it's absolutely fascinating, but how practical this is and relevant for growers to use as a tool, um, has just been absolutely fascinating. Increase in yield 20% is a lot when it comes to an improvement you can make in a cultivation space. So, um, yeah, I mean, having your airflow minimum of 200 feet per minute is the huge takeaway here for a grower, because everything goes back to that. For me, it's even more important than nutrition, because this is actively taking that nutrition up. It's really a steering tool.
Like
Um,
allowing for that absorb, like it doesn't matter what you eat if you can't absorb it, right?
yep,
So it's like
exactly,
you should be shoving food in your face. If you can't digest it, it's not going to do you any good. So if you,
exactly,
you can pump nutrition into the plant substrate, but if it's not going to drink it if it.
exactly, exactly.
So that makes me think again, like would that work for folks that have stacked systems because they've only got so much height that they can work with
Hmm.
before, the plants get too tall for that short shelf.
Hmm.
So that's the one thought that I have. And then the next thought that I have is it sounds to me like that is better imitating growing outdoors if you've got because outdoors, I mean, it's not always windy, but it's
Right,
not the same as, when you have a fan on in your house, you know?
exactly.
And we know that outdoor growers grow larger plants and get higher yields and all of that stuff.
Hmm.
So do you feel like that has thinking like a plant?
Yeah, thinking like a plant. Yeah, absolutely, absolutely. It might not be all the time, but there is airflow outside, like
Mm-hmm.
constantly, right? It might be hard for 1 second and soft for the next, but it's always there and it's always breaking that boundary layer. So thinking about it from that standpoint, yeah, I mean, it definitely takes it to the next level. And at this point, when I go visit facilities, I always have my anemometer with me where I can measure airflow because, I'm finding not only are people not putting enough air in their system, and when I say taller, I don't mean taller to extent of it being
10 feet.
like yeah, like I don't mean negative taller, right? It's certainly something to think about, if you're in a vertical stack limited system, but there's other techniques you could do, ramping up your lights faster, for example, to keep your plants a little short. There's other techniques you can kind of
Yeah.
combine with it, that you might have to play with once you get your system working. But the amount of people that in 90% of their areas have 0% airflow is much higher than those who have adequate airflow.
Hmm.
So it's whether it's stacked or whether it's stacked or not, whether you're using PIP's actual system or not, it's a factor to take into account if you're a grower.
Hmm.
Just knowing your air velocity. It's right up there with you. Do you have your par meter or do you have your anemometer? Because um, if you've got a fan system where it's just a fan on the side of the wall, and so the plants right by it are getting a ton of airflow. The ones, 5 feet down, it's nothing. Those are going to drink differently. If these are all the same variety and you're wanting to water them the same, and then half of them are drinking differently, you're going to have, two different crops there.
Oh man, I
So,
feel like I could have. used this information a couple of years ago when we were getting like tall plants and then short plants in the middle, but we had the fans that were around the outside, and
Yep.
it was kind of like a racetrack of air going
Hmm.
around, and we were trying to figure out like because the floors weren't exactly level because it was, hoop houses, whatever.
Right.
And so there were like dips and hills and stuff, and we were like, okay, is water pooling somewhere? And like, but that makes a lot of sense that it would have been the airflow, and they're drinking
Hmm.
differently and they're uptaking more nutrients when they're drinking more and it's getting moved through the plant more. That's super interesting. And I think that's the kind of stuff that can really be useful for cultivators, you know,
Yep.
which is awesome because that's what we want more of.
That's right. That's right. If the plants aren't growing well or efficiently and the farmers can't be successful, everyone downstream is in trouble too. So all
Mhm.
the way to the patient. So, I come from a fourth generation farming family. So, I don't know. I feel like my mind is just made to, think what can we do in science that's going to be something practical, but still
Mhm.
interesting and scientific that the farmer can take and walk away with to be successful.
Yeah, because
Yes, that's what excites me.
that's definitely not something I ever thought of, you know. I mean, airflow
Yeah,
obviously is important to keep those microclimates from forming, which you can have other issues with pests and pathogens.
right.
You want to keep your air flowing, but I didn't, yeah, the transpiration thing. That didn't even cross my mind. I wasn't thinking like
Yeah.
a plant.
Yeah, no, I mean, sitting back now, it may even for
Yeah,
myself, it makes sense, but at first, I wasn't quite thinking of it that way and how big of an impact it'd be.
that's cool. I
Yeah,
feel like, the research that you do is incredibly valuable to not just, cultivators, but, science for science sake, but also like the consumers and letting people know like what's going on. Um, and you're doing so much cool stuff. And so you should tell people like how they can participate, if they can participate in any of your research, how they can participate, like what kinds of studies you're doing, especially, when we're talking about like figuring out zones for various types of cannabis and where they grow well and where they don't. And like where they can find you, how they can contact you, where they can sign up for various studies, what you got going on, where they can find your research and all that good stuff. Because
absolutely. So for the hemp mine, if you are in need of hemp varieties or hemp goods, you can find us at thehempmine.com.
and the hemp mine is one of the few places where you can get hemp clones, is my understanding. Like
Yes,
most hemp comes as seed, but
right.
the hemp mine offers clones, which is super cool.
Correct. That's right. And then for everything cannabis research, it's free for growers, for researchers, for students. It's open source. We ask people to sign up just where we're not getting bombarded with bots and that sort of thing. But once you sign up, it's just a check mark, and then you're allowed to get on our website. And there's forums where people can share information and thoughts. There's our SOPs. There's videos from gosh the last 3 or 4 years from everything from what to do about fire ants to gosh lighting anything under the sun cultivation, but that's where you would sign up, and then if we have new studies coming out or any sort of collaboration participation open forum calls, you'll get emails of how you can join and participate. So the CRC website is cannabisrc.org. You can also check us out on Instagram. My handle is dr.justice, JUSTICE _ grows. And then, of course, CRC research, because we can't have cannabis in the Instagram name.
There you go. So I'll pop up those links for any viewers who want to participate or join or sign up for newsletters or whatever, the case may be. Um, but dude, awesome conversation. I'm so glad I got to finally talk to you.
Me
And
too. Me too.
I really hope that I get to see you at a conference soon. Yeah, but I'm sure we'll come across each other in person at some point, which will be awesome. But it was so good to
Absolutely.
have you on the show. And again, it's a pleasure to meet you. And your research is fascinating and amazing. Thank you so much for
Thank
sharing.
you.
All right. Well, I will talk to you later, Allison.
All right. Thank you so much. I look forward to seeing it.
All right. Bye.
See ya.